All right, so this is our fluid balance and RBCs lab, and it's a continuation of our exploration of osmolarity, and this time we're looking at it in living cells. So, the cell membrane of an RBC, or most of your cells, is permeable to water. Water is just a tiny, even though it's polar, it's so tiny it can just blast through the membrane from one side to the other. So the cell membrane is permeable to water, but most other molecules, as we're going to talk about, have trouble going across the membrane without particular help. It is going to be important for this lab, though, to think about whether or not a particular molecule can cross the membrane or not. Particularly things that are lipophilic, are going to be able to get into the hydrophobic part and just end up on the other side of the membrane without any particular channels. Whereas other things, polar molecules like sucrose or something, what's going to happen when that comes up to that hydrophobic region? It's just going to want to stay in the polar areas where all the water molecules are. So part of this lab, when you're developing your hypotheses, you really need to pay attention to whether the solute molecule in the solutions that we're putting the cells into, whether those solute molecules are going to be penetrating, meaning they can cross the membrane without any help, without any carriers or channels, or whether they're non-penetrating, which means they hit the membrane and they can't cross. So the basic idea is we've got RBCs, red blood cells, which we are going to take. We'll take like 6 drops of red blood cells, and we're going to add them to some solution. We're going to have 3 mils of some solution. We have different types of solutes. We have different concentrations of different solutes. When you think about what are our independent variables in this experiment, that's kind of the clue there. Got a couple of things going on that we are varying. And then we're going to put them in a test tube, and then we want to know what happens. And there's a few things that could happen. It could be the cell just hangs out and is happy, or water might move into the cell, or water might leave the cell. For each of the 9 test tubes, you need to come up with a hypothesis before you start the lab. And to come up with a hypothesis, you need to think about 2 important things about the solution that the red blood cell is going into. Over here, you've got to think about the osmolarity of what is the osmolarity of the solution that the red blood cell is heading into. You have to calculate it. Again, calculating that's going to depend on whether or not the solute it dissociates or not. So you make sure you understand the relationship. Molarity and osmolarity are related, but not necessarily the same thing. And then you also have to make sure you remember a blood cell in most cells, most of your physiological fluids have an osmolarity of around 0.3 osmolar, or you could also say 300 milliosmolar. Obviously, those are the same thing. So that's one thing. For each of the solutions, you have to think about osmolarity. But another important thing is you have to think about whether the solute is penetrating or non-penetrating. And between these 2 pieces of information, you're going to be able to make a prediction on what's going to happen. Which way is the water going to cross the membrane due to osmosis, and which direction is it going to go? So, we have these words. Crenation means kind of shriveling up. If water is leaving the cells, it gets more shriveled up. That would be crenating. Water might come into the cell, in which case it starts swelling. If it swells enough, think about blowing up a balloon. A balloon gets bigger and bigger. You can actually have the cell explode. That's hemolysis, basically lysis, breaking apart of the blood cells. In that case, the cell is going to just spill out. Remember, every blood cell's got 250 million molecules of hemoglobin inside. and all that hemoglobin is just going to go into solution. It's no longer going to be contained within the cell membrane of the red blood cell. So you can actually use that fact to come up with our assay. Over here, how do we figure out what happened to the cells? So we're going to take our blood cells and put it in a solution, mix them up. It's going to look red because it's a suspension. You got red blood cells suspended in whatever solution. But then we're going to put it in a centrifuge, and Tina's going to talk a little later about the centrifuge. But it's basically spinning, spinning, spinning. That's why I did a little spinning thing. Basically, really high G's, like, whoa, the gravity pulling down gets really strong. If you have cells that are still intact, the cells will all just get pulled down to the bottom of the tube. We call this the pellet. And the solution is just going to be still up here, as we call the supernatant. So if the cells don't lyse, then the cells just get pulled to the bottom after you've been spinning it. You can see that. If the cells do lyse, though, then the hemoglobin is just free in solution. It's no longer contained in the blood cells, and no matter how much you spin it, once you just dissolve these little hemoglobin molecules in the solution, they're just going to stay in solution. So even after spinning, the supernatant is just going to stay bright red. So that would be an indication that hemolysis happened. It doesn't tell you, hemolysis doesn't mean, hypotonic could just be inflating, doesn't necessarily have to lyse. And just because you have a pellet here and we know the cells didn't lyse, doesn't mean the cells were necessarily happy. They might have crenated. One thing you might want to look at when you look at your little tubes is compare the size of the pellet in different situations. One thing I like to think about grapes. A grape, you could have 20 grapes in a bowl and they're going to take up a certain amount of volume. And a raisin is just a dehydrated, shriveled up grape. If you take 20 grapes and turn them into 20 raisins, they haven't broken apart or disintegrated, but they take up less volume. And you might see a similar thing if you're comparing kind of isotonic cells to hypertonic cells. So it's worth looking to see if you notice something like that. But that's the basic background. But again, before you start this, you need to come up with a hypothesis. We have provided a chart here where for each solution, you're going to have an opportunity to calculate the osmolarity, really think explicitly whether it's a penetrating or non-penetrating solute, and then make your prediction about what you think is going to happen. You can also just write it as, it doesn't have to be on the chart, but you have to always have that information for each of the situations. What is the osmolarity? And you have to calculate it. It's a little bit of work, and then make your prediction. When you're done and finally get all your data, you're going to want to make sure you record what happened. And in this case, it's worth recording a picture too. Like when you're taking your lab practical later on in the semester, it's not just about the kind of intellectual content. It's about the actual practice. How did you actually know? Like on your lab exam, you might have a test tube that you look at and you have to decide what happened here. What does it mean? Interpret what happened to these blood cells. And if you just kind of scribbled some words like hemolysis, no hemolysis, and you're not going to remember 2 months from now, how did I even know that? So make sure when you're writing down your data, describe things more completely. Describe the actual look of it and how you use that to actually make your decision what happened to the blood cells. So what we're going to do now is actually go over to the lab bench, and we'll show you how to actually set up the experiment.